In genetic males, the presence of the Y chromosome is the defining factor for male sexual development. The Y chromosome carries the SRY gene, sex-determining region Y gene, which acts as a master switch. This gene produces a protein known as the testes determining factor, TDF. If this gene is active, the indifferent gonads of the embryo are signaled to develop into testes instead of ovaries. The two crucial cell types in the developing testes, Sertoli cells and Leydig cells. Sertoli cells are the supporting cells within the seminiferous tubules. Starting at around 7 weeks of gestation, they secrete Müllerian-inhibiting substance, MIS, also known as anti-Müllerian hormone, AMH. This hormone acts locally to cause the regression of the Müllerian duct on the same side as the testis secreting it. Leydig cells are located in the interstitial space of the testes. Starting at around 8 weeks of gestation, they begin secreting testosterone. This testosterone production peaking between 15 to 18 weeks of gestation, which is crucial for masculinizing the fetus. Müllerian-inhibiting substance has two primary jobs. It degrades the Müllerian ducts in males. It plays a key role in the first phase of testicular descent, facilitating the movement of the testes from the upper abdomen down to the pelvis. Because MIS causes the Müllerian duct to regress, it does not disappear entirely. Small, non-functional structures remain in the adult male as remnants of these embryonic structures. 1. Prostatic utricle, a small, blind pouch located in the prostatic urethra, homologous to the female uterus, vagina. 2. Appendix of testes or hydatid of Morgagni, a small remnant tissue on the upper pole of the testis, homologous to the fallopian tube. Testosterone is actively promoting and stabilizing the Wolffian ducts, which are the precursors for the male reproductive tract, such as seminal vesicles, epididymis, ejaculatory duct and vas deferens. To develop external male structures, such as the penis and scrotum, testosterone itself is not strong enough. It must be converted into a much more potent androgen called dihydrotestosterone, DHT. The enzyme responsible for converting testosterone into the highly potent DHT is 5-alpha reductase. A deficiency in this enzyme leads to ambiguous external genitalia at birth because the external tissue cannot process the testosterone into DHT. The default path for human embryo development is female. Without a Y chromosome, the primitive gonads automatically turn into ovaries. Testes make anti-Müllerian hormone, AMH, to destroy these female ducts. Because there are no testes and no AMH in a female embryo, the Müllerian ducts survive and mature into internal reproductive organs, such as fallopian tube, uterus, cervix, and upper 2/3 of vagina. Lower 1/3 of vagina is derived from sinovaginal bulb of urogenital sinus. Because females lack Leydig cells, the Wolffian ducts wither away, leaving behind useless leftover tissue remnants, such as epoephorin, parophorin, and Gartner's duct. The leftovers, epoephorin and parophorin, end up tucked inside the broad ligament, which is the tissue sheet holding the uterus in place. Sometimes the Gartner's duct is present along the entire length, leading to blockage forming Gartner's cyst. Epoephorin and parophorin can also be blocked, forming paratubule cyst. Initially, Müllerian duct was lateral, then it grows in above-downward direction. While growing downwards, they cross the Wolffian duct and they meet with the Müllerian duct of other side and fuses. Form a single uterus and upper vagina. By week 10 of pregnancy, an ultrasound or microscopic check can visibly distinguish whether internal organs are male or female. The primitive uterus structure starts forming at 10 weeks, but its hollow inside chamber, that is uterine cavity, is completely hollowed out and finished by week 20. While males need hormones DHT to grow external parts, the female external parts form automatically without needing estrogen inputs. Indifferentiated external genitalia have 3 structures. 1. Genital tubercle. In males, the embryonic genital tubercle develops into the penis. In females, it develops into the clitoris. 2. Genital swelling. In males, the embryonic genital swelling matures and fuses to become the scrotum. In females, it develops into the labia majora, the outer lips of the vulva. 3. Genital fold. In males, the embryonic genital folds fuse along the midline to form the penile urethra. In females, the embryonic genital folds remain unfused to become the labia minora, the inner lips of the vulva. Let's clear some cysts related to this topic. 1. Gartner's cyst is found on the front, that is anterior wall of the vagina. 2. Bartholin's cyst forms in a different gland located on the back, that is posterior entrance. 3. Paraurethral skein cyst is just adjacent to the urethra. 4. Inclusion cyst is the most common cyst in vagina or vulva. It is the most frequent non-cancerous lump found, usually caused by surface tissue getting trapped under the skin during healing or childbirth trauma. Thanks for watching. Subscribe for more updates.